Hey Team Bio, Chloe just asked me why I came up or how I came up with that tagline. I don't know, just started saying it. Now I say it without even thinking about it. Okay, so today's screencast is going to be an introduction to errors in meiosis and their results. So we're going to be discussing specifically Down syndrome and how it occurs. Okay, so take a look at these two different karyotypes. Remember, a karyotype is pairs of homologous chromosomes. So these are homologous chromosomes. And homologous chromosomes share all the same genes, but they have different alleles, different sequences at those genes, because a gene is a location on a chromosome and an allele is the sequence of base pairs. So if this is karyotype of individual number 1, we see they have a chromosome number 1 that they got from their biological mother and a chromosome number 1 that they got from their biological father, and so on and so forth down the length of all the pairs. They have 23 pairs total for 46 chromosomes in total. So what we call their diploid number, when we add up all of their chromosomes, we see it is equal to 46. So notice here that this individual has 2 X chromosomes and there's nothing where the Y is. Now, this is an individual who would be a biological female for their sex. We are not going to talk in a lot of detail about secondary sex characteristics and how people develop male or female genitalia and intersex. But if you're interested, I can tell you more about that. But for right now, we're just going to keep it like pretty basic in terms of the genetics and the phenotype. An individual who is XX would be, their sex would be a biological female, and an individual who is XY is a biological male. Okay, so notice that, so this person is XX and this person is XY, but another big difference between karyotype number 1 and karyotype number 2 is if you look carefully at all of the pairs of homologous chromosomes, you'll see that in karyotype number 2, this person has 3 copies of chromosome 21. And then we call this a trisomy. Anytime that you have 3 copies of a chromosome, it's called a trisomy. If you're missing a copy of a chromosome, it would be called a monosomy. So this person's diploid number is equal to 47. And they have the phenotype of Down syndrome because they have an extra copy of chromosome number 21. We're going to talk in a second how this happened. It's the result of nondisjunction, likely in an egg, but it also could happen in sperm production. And we see the most common trisomy is trisomy 21, that you also see individuals sometimes that have trisomy number 18 or trisomy number 13. For many of these chromosomes, a trisomy would result in a spontaneous miscarriage of the developing embryo, because having 3 copies of, for example, chromosome number 1 is not compatible with being alive. Most trisomies result in spontaneous miscarriages of the pregnancy, but 21, 18, and 13 are examples of chromosomes where you can have a trisomy and you can have a a happy, healthy human. Okay. So, non-disjunction, how does it happen? First of all, let's break down, or actually I shouldn't say necessarily healthy. People who have Down syndrome have often have enlarged hearts and can have health problems as a result. They also have intellectual disabilities, and same with 18 and 13. They do lead to significant health impairments, so I shouldn't say that they are necessarily healthy. Okay, non-disjunction. What does this mean? Let's break this word apart. So a junction is two things joining or coming together. So for example, White River Junction is the place where the White River comes into the Connecticut River. So let's say this is a joining. A disjunction would be breaking apart. And a non-disjunction, this entire thing, is a failure to break apart. Which I guess is kind of the same thing as a junction, but in non-disjunction, the intention is to have a disjunction and that breaking apart somehow fails. Okay, so you know in a normal meiotic division, you start with homologous chromosomes lining up at the metaphase plate in metaphase 1 of meiosis, and then during anaphase of meiosis 1, the homologous chromosomes separate. We should write that down. And then in meiosis 2, sister chromatids separate. Remember, sister chromatids are the result of DNA replication. So every sister chromatid is a clone of the other sister chromatids. In meiosis, this isn't quite true because during prophase, we have crossing over. So we end up with these chimeras, these mixtures of DNA that you got from your biological mother and DNA that you got from your biological father on these sister chromatids. But anyway, sister chromatids are separating during anaphase of meiosis 2. And then you produce 4 gametes, or gametes each with the total chromosome number of N. So this is called a haploid cell. And when a haploid cell joins with another haploid cell, then you get the 2N, which is our diploid number right here, 46. So in a human, we would see 23 chromosomes in each of the gametes, and when an egg fuses with a sperm, we would get a total of 46. Okay, so what happens when we have a non-disjunction, a failure of the chromosomes to break apart? Well, we have 2 scenarios here, and they are non-disjunctions that happen in different phases of meiosis. So if you have a non-disjunction, the failure of the homologous chromosomes to break apart during meiosis 1, that means both of these chromosomes are pulled to the same side of the developing embryo. Or the developing, sorry, not an embryo. It's not an embryo yet. These are the developing gametes here, either egg or sperm. So there's a failure of these chromosomes to break apart in the center here. So you're left with, in meiosis 2, one of the products has an extra chromosome in it, and one of the products is missing a chromosome. So for example, if this was chromosome number 1, there would be, there are 1, 2, 3, 4 sister chromatids in meiosis 2, and this is totally missing any sister chromatids of of chromosome number 1. So ultimately, you end up with a gamete that 2 of the gametes have an extra copy of chromosome number 1, and 2 of the gametes are entirely missing a copy of chromosome number 1. Okay, so if you had a fusion of this gamete with this gamete, you would end up with an individual that has a trisomy. It would have 1, 2, 3 copies of this large chromosome. If you had a fusion of this gamete with this gamete, you would be entirely missing 1 copy of chromosome number 1. So this would be called a monosomy. Okay, so that's if you have a non-disjunction, a failure of the chromosomes to break apart, the homologous chromosomes to separate during meiosis 1. You have 2 gametes with an extra chromosome and 2 gametes missing a chromosome. What about meiosis 2? Well, if you have a proper division of the homologous chromosomes during meiosis 1, one side of the developing embryos will have the standard number of chromosomes, or N, 1 of each copy of every chromosome, and a non-disjunction in meiosis 2 will lead to 1 gamete with an extra chromosome, so 2 copies of chromosome number 1, and 1 gamete entirely missing a chromosome number 1. So similarly, if you have a fusion of the chromosome with an extra with a standard gamete, then you'd have a trisomy, and if you have fusion of this one with one of these products of normal meiotic division, then you would have an individual with a monosomy. Okay, that is all on non-disjunction and its results. Okay, bye.